Hello everyone, my name is Agastya Sarmah and I'm a first year student at the University of California, Berkeley studying molecular and cell biology. Now for my project, I focus on liver cancer, the third leading cause of cancer-related deaths worldwide. More specifically, I studied hepatocellular carcinoma, HCC, the most common form of liver cancer. What makes HCC particularly interesting is that it exhibits striking disparities across race, gender, and ethnicity, highlighting the need to understand how demographic factors shape tumor biology. In my study, I investigated ARID1A, a gene frequently mutated in HCC. ARID1A mutations disrupt chromatin remodeling and DNA repair mechanisms, and my goal was to determine whether ARID1A mutations contribute to HCC disparities and if these mutations can be targeted therapeutically. I analyzed genomic data from 2,351 HCC patients across 14 studies on CBAL portal and compared survival outcomes between patients with and without ARID1A mutations. Patients with ARID1A mutations had a median survival of 23.46 months compared to 53.29 months for those without the mutations. But the data was not evenly distributed. Black patients showed the highest prevalence of ARID1A mutations compared to white and Asian patients. Moreover, Black patients had the lowest median survival at 9.83 months versus 27.17 months for white patients and 38.47 months for Asian patients. To expand on these findings, pathway analysis revealed that tumors from Black patients exhibited enriched alterations in adaptive and innate immune pathways, along with lymphocyte depletion, likely weakening the immune response to cancer. To explore whether certain gene mutations might be disadvantageous to cancer cell survival when co-mutated with ARID1A, thereby conferring a survival benefit to patients, I performed a virtual synthetic lethality screen. Specifically, I examined genes that were co-mutated exclusively in disease-free ARID1A mutated HCC patients who did not experience recurrence after treatment. I compared those genes to the co-mutated genes found in deceased patients, and my analysis identified 26 unique genes, with POLQ being the most frequently mutated. What makes POLQ particularly interesting is its role in DNA damage repair, similar to ARID1A. Patients with POLQ mutated HCC had a median survival comparable to those with ARID1A mutated HCC. Notably, patients with mutations in both ARID1A and POLQ exhibited prolonged survival with no adverse outcomes. This suggests a synthetic lethal relationship between the two genes. Expression analysis revealed that ARID1A and POLQ are co-expressed in healthy liver tissue. However, this correlation is disrupted in tumor tissue, hinting at a compensatory mechanism between the genes. Ultimately, my findings position POLQ as a promising therapeutic target in ARID1A mutated HCC. Inhibiting POLQ in these tumors could selectively impair cancer cell survival while sparing healthy cells with functional ARID1A, which can potentially lead to more effective and personalized treatment strategies. Thank you so much for your time, and I hope you have a wonderful day.